Dear students, moving further to the flight test, in the previous discussion, we discussed about experiments on moving bands of locust or the moving swarms of locust. Today, I would be discussing certain experiments on flying individuals in a swarm of locust. Kennedy in 1951 performed experiments on flying individuals in a swarm, changing the sun's image from the right to the left side by a large mirror as locusts flew through the shadow of a tree led to a turnaround of the animals, continuing flight in opposite direction as long as they were in the tree's shadow and exposed to the reflected sun. As a note of caution, it should be mentioned that these experiments could be performed only on 5 animals. In addition, migrants, when thrown off from their course by disturbances, reverted to their original course soon after, even if isolated from the migrants. Now, this is another thing that this experiment was conducted on only 5 organisms, and the migrants, specifically if a 1 or 2 or 3 number, they are disturbed from their course, they revert back to their original course even if they are isolated from the group or the swarm. So these again are results based on experiments, so definitely are difficult to describe. Taken together, these experiments by Kennedy, it provides evidence for a sun compass orientation in larval and with some reservation in adult migrants, although no quantitative data were collected. The wing stroke of locust is remarkably constant and independent of external conditions. The flight behavior of the desert locust, that is Cystosarca gregaria, was studied under various experimental conditions in order to find which external factors can initiate, maintain, or alter the wing movements, excluding reactions which depend upon higher nervous centers. The tarsal reflex and the response seen when the aerodynamic sense organ on the head is stimulated. This was suggested by Westphal. These were reinvestigated in order to relate them to two hitherto unknown reactions: the maintenance of flapping when the wings are exposed to wing, and the regulation of the lift when the body angle, that is angle of pitch, is changed during forward flight. Inhibition. As in most other insects, the flight of a locust cannot be started when the legs or only part of one leg have contact with the rigid body. Flight stops when such contact is regained. Amputation of the legs abolishes these reactions, so showing that some leg proprioceptors inhibit flight. So it is actually because these insects, or insects as an organism itself, they have walking legs as well as they have wings. Now the flight in these insects, so especially related to the migratory insects and locusts, has been observed that egg has some the leg has some proprioceptors which receive and perceive, and some receptors, some sort of signal which inhibits flight initiation. A suspended locust can be induced to fly in 3 ways. Number one, by application of a sufficiently strong stimulus which normally provokes escape reactions. The flight lasts only for a few seconds and adaptation is generally very quick. By sudden removal of the support for the legs, that is the tarsal reflex, although not confined to the tarsi, but it is termed as a tarsal reflex. The flight lasts 5 seconds on average, corresponding to 100 wing strokes. There is practically no adaptation. By blowing upon the wind sensitive hairs on the head, the wind must exceed 2 m per second, but its direction is of little importance. Since the static bending has no effect, the adequate stimulus seems to be minute vibration of the hair. The flight lasts as long as the wind blows, and the hairs are therefore also involved in the maintenance of flight. So, flight, of course, is related to the proprioceptors of the leg. It is related to the tarsal reflex, and also the wind sensitive hairs which are present on the head of the locust. It has been suggested by the uh, workers who have studied the flight mechanism or flight in locust. When locust has stopped, the legs begin to flutter and eventually remain still, but normally flight is not resumed unless one of the above stimuli is applied. Two receptor systems are involved. Number one, the wind sensitive hairs on the head. In a wind, they emit impulses irrespectively of whether the locust has any chance of flapping its wings or not. Wind on the head is therefore an extrinsic flight stimulus. The flight posture is never complete. Second, a hitherto unknown receptor system in the pterothorax, which was studied in insects whose supraesophageal ganglia was cauterized. It maintains the movements when the wings oscillate in a wind but cannot initiate them. The adequate stimulus is the rhythmically changing wind pressure on the wings. Wind on the wings is therefore an intrinsic flight stimulus. So there are two flight stimulus. One is the extrinsic, which is because of the wind on the head, and the other is intrinsic, which is because of the wind on the wings. When the average lift exceeds half the body weight, flight continues in complete flight posture but stops when the lift approaches zero. The experiments indicate that the stimulation ceases when the lift becomes negative during the upstroke. The receptors are unknown. It is suggested that they are situated at the wing hinge, at the joint. The locust does not adapt to either of these stimuli and invariably stops a few seconds after they have seized control of lift. The locust tends to keep the lift constant during a given performance, and this observation, together with the constancy of most stroke parameters, made it possible to investigate the mechanism involved. The method was to make the insect fly steadily against a horizontal wind and then alter the inclination to the wind, which is equal to the body angle, at regular intervals. The data permitted an estimate of the mean change in wing twisting. The triangle theta. Triangle theta increases wings pronated by 15 plus minus 3 degrees when the body angle was increased from 0 to 15 degrees. This is the main factor in the control of lift. Now these are all experimental work which have been done for the related to the flight in locust, and the angles have been worked out. The inclination of the wing in relation to the wind has been worked out, and has been described here. The discussion shows that this presupposes a system of lift sensitive receptors, and probably these are the campaniform sensili at the wing hinge. If present in other insects, the homeostatic character of the wing stroke of Drosophila may therefore be caused by a nervous mechanism and need not be a consequence of the energetics of flight. Now this is for the Drosophila. Central rhythm. It is concluded that the central nervous system does not initiate flight rhythm on itself, that is de novo, does neither determine the stroke frequency nor the strength of the contractions of the controller depressor muscles, may control the phasing of the contractions, although a simpler hypothesis is proposed or advanced. and may control the indirect flight muscles, but only as far as to produce stimuli of constant strength. And this constant strength could be what can be termed as the maximum strength. Thank you. So maybe you might not find this very interesting uh, right now, but in the forthcoming discussion, you would really like to study the flight mechanism in locusts.